Hello friends, in this clip we shall discuss about few questions on photosynthesis. Now let's see some basic concepts in photosynthesis. Now photosynthesis takes place in organelles called chloroplasts. Each chloroplast, now this is structure of chloroplast. Okay. Now each chloroplast is covered by two membranes here. You can see outer membrane and inner membrane. Now these two membranes they enclose a fluid matrix which is called as stroma. Now this stroma has many flattened structures. You can see these flattened structures here. So stroma has many flattened structures. These structures are called thylakoids. Now thylakoids, this is one thylakoid, second, third, fourth, like this. Okay, so thylakoids they are placed one above the other to form one granum. Now this is one granum. So many thylakoids they are placed one above the other to form one granum. Like this, you'll see many grana here. Okay, and the grana, okay, they are interconnected. Here you can see this is one grana, this second grana, this is one more grana. Okay, now these grana they are interconnected. Here you can see they are interconnected by lamellae. Now these are all lamellae. Okay, so lamellae they interconnect the grana. Now, these grana or thylakoids, okay, they are the sites of photosynthesis as pigment molecules are found on them. Apart from grana and lamellae, apart from grana and lamellae, there are ribosomes. Here you can see ribosomes are there. Okay, starch granules, lipid droplets, and also a circular DNA. So all these are the components of chloroplast. Okay. On the surface of thylakoids, light harvesting complexes are present, which are important for photosynthesis. So light harvesting complexes are found on the surface of thylakoids. Okay. Now remember that these thylakoids they are the main sites of photosynthesis, the light reaction. Okay, so the components which are involved in light reaction of photosynthesis, they are all present on these thylakoids as well as on the lamellae. Now, light harvesting complex. Okay, so these light harvesting complexes they are found on the surface of thylakoids. Now, they are also called antennae. Now, this is light harvesting complex. Okay, these complexes are also called as antennae as they trap the sunlight which is necessary for photosynthesis. Now, each light harvesting complex is made up of several pigment molecules. Here you can see. Now, this is the light harvesting complex which is made up of many pigment molecules. Here you can see these are the many pigment molecules which absorb the photons, okay, which absorb the photons or energy packets from the sunlight. Now these pigment molecules include xanthophyll, carotenoids and chlorophyll. They are the major pigments. As these pigment molecules absorb the photons, they get excited and they release the electrons. These electrons from different pigment molecules are absorbed by reaction center. They are absorbed by reaction center. Okay. Now reaction center consists of chlorophyll molecules. Okay. The electrons released from the reaction center are accepted by an electron acceptor. Okay, the primary acceptor. Okay. As the electrons are lost from the molecule, okay, it gets oxidized and molecules that accept the electrons get reduced. So it means, so suppose if the electrons they are accepted by the reaction center chlorophyll, when this reaction center loses the electrons, okay, so this molecule is said to be oxidized and primary acceptor that accepts these electrons, it is said to be reduced. There are two photosystems here which are present, okay, on the thylakoids and also on the stroma lamellae. There are two photosystems. Okay. Each photosystem is made up of its own light harvesting complex. Photosystem 1 is located on both thylakoids as well as the interconnecting lamellae. The reaction center of photosystem 1, it absorbs light at 1700 nanometers wavelength. Means maximum absorption of PS1 occurs at 700 nanometers wavelength range of the light. Whereas photosystem 2, it is located only on thylakoids. Remember, photosystem 1 is located on both thylakoids as well as lamellae, but photosystem 2 is located only on thylakoids. And the reaction center of PS2, that is photosystem 2, it absorbs light at 680 nanometers peak wavelength. Okay, means it is the uh, at this wavelength the photosystem absorption is maximum. Absorption of light by this photosystem is maximum at 680 nanometer wavelength range. Now, stages in photosynthesis. There are basically two stages in photosynthesis, namely light reactions and dark reactions. Okay. Now, light reaction occurs in grana and lamellae. Grana which consists of thylakoids, and also lamellae which connect the two consecutive grana. Now, light is required in this process. That is what is called light reaction. During light reaction, ATPs are synthesized. Okay. During light reaction, ATPs are synthesized, and also NADPH molecules are also synthesized. Now, light reaction is divided into cyclic and non-cyclic phosphorylation that we shall see later. Now, dark reaction. It occurs in stroma. Light reaction occurs on thylakoids and lamellae, whereas the dark reaction occurs in stroma. Okay. Now, it is light independent. It means light is not necessary for dark reaction to take place, but for light reaction, light is compulsory. Now here, during that reaction, what happens? ATPs are used to synthesize carbohydrates. That is why dark reaction is also called as biosynthetic phase, because biomolecules are like biomolecules, carbohydrates are synthesized. Okay. Light reaction involves cyclic and non-cyclic photophosphorylation. Now here, phosphorylation means formation of ATPs. Photophosphorylation means formation of ATPs in the presence of sunlight. Now let's see cyclic photophosphorylation. Okay. Now cyclic photophosphorylation involves photosystem 1. Okay, so this chlorophyll P700. Okay, it is a reaction center of photosystem 1. Okay, so cyclic photophosphorylation takes place only, okay, it involves only photosystem 1. Okay, right. Now here the light harvesting complex absorbs the sunlight. So this is the light harvesting complex. Okay, reaction center of the light harvesting complex. Okay, it absorbs the sunlight, gets excited, and releases the electrons. So once this chlorophyll P700 molecule, that is reaction center of photosystem 1, okay, it absorbs the sunlight, it gets excited, and it releases electrons. It releases electrons. Now these electrons which are released, they are accepted by an electron acceptor. Now when this electron acceptor is oxidized, the electrons pass through an electron transport system, and electrons are returned to the reaction center. Now it is called cyclic photophosphorylation. Okay, because the electrons, they return back to the reaction center. So they are first lost from the lost, or you can say they are released by the reaction center. Again, they come back to the system. Okay, or they come back to the, they are received back by the reaction center. That is why it is called cyclic photophosphorylation. Now in this process, ATP is synthesized from ADP. So in cyclic photophosphorylation, only ATP is produced. Okay. Here you can see in cyclic photophosphorylation, only photosystem 1 is involved. But in non-cyclic photophosphorylation, this is non-cyclic photophosphorylation, here both photosystem 1 and photosystem 2 are involved. Light harvesting complex of photosystem 2 absorbs sunlight and releases electrons, okay, which are accepted by an electron acceptor, and this electron then passes through the electric transport chain and reach the light harvesting complex of photosystem number 1. Now, light harvesting complex of photosystem 1 also absorbs sunlight. It also absorbs sunlight and releases the electrons which are accepted by an electron acceptor. Now, these electrons trans are transferred from electron acceptor to a molecule of NADPH. So the electrons released by electron acceptor, okay, they are accepted by NAD plus, NADP plus. As a result, NADP plus is reduced to NADPH. Okay. So in non-cyclic photophosphorylation, both ATP as well as NADPH are synthesized. Now, since the electrons do not return back to the reaction centers, now this is called non-cyclic photophosphorylation. It is also called as Z scheme. So electrons here they never return to the reaction centers, but they are finally accepted by NAD plus, NADP plus. Okay, but in cyclic photophosphorylation, these electrons they return back to the system. Now, another important event occurring during the non-cyclic photophosphorylation is splitting of water. Here you can see splitting of water and release of oxygen. Okay. Now, evolution of oxygen. Okay. Now, a water splitting complex consisting of manganese and calcium. Water splitting complex consisting of manganese and calcium splits water to release protons, electrons, and oxygen. Now, this water splitting complex is also called as oxygen evolving complex because it ultimately results in evolution of oxygen or release of oxygen. Remember, water splitting is associated with non-cyclic photophosphorylation where water is split into protons, electrons, and oxygen by a water splitting complex or oxygen evolving complex. This water splitting complex is made up of basically manganese and calcium. Chlorine is also essential for the water splitting. Next, chemiosmosis. Now, chemiosmosis, okay, is a process in which ATPs are synthesized from ADP. So, chemiosmosis is nothing but the process of synthesis of ATP from the ADP, okay, with the help of an enzyme called ATP synthase. Okay, so ATPs are the end products of chemiosmosis. So, in a process called chemiosmosis, ATPs are synthesized with the help of an enzyme called ATP synthase. So, this enzyme is found on the membrane of thylakoids. Now, this is the membrane of thylakoids. So, on the membrane of thylakoids, you find ATP synthase, and this is the lumen of the thylakoid. Now, this is the ATP synthase that is found on the membrane. Now, ATP synthase has two parts. Okay, one is called FO, second part is called F1. The protons, okay, these protons, hydrogen ions, the protons resulting from the events in non-cyclic photophosphorylation, such as splitting of water, they get accumulated in the lumen of thylakoids. Now, the concentration of these protons within the lumen is high. Okay. As a result, the protons they start diffusing from lumen of the thylakoid into the stroma. Okay, and when they diffuse through the enzyme that is ATP synthase. Now, during this process, ATPs are synthesized from ADP, and these ATPs are utilized in dark reaction of the photosynthesis. Now, this is all about light reactions. Now, let's discuss about dark reactions. Now, reactions they are called so because they are independent of light, means they don't require light. Okay. Now, these reactions take place in stroma of the chloroplast. Now, dark reactions are completed in a cycle called as C3 cycle or Kelvin's cycle. It is completed in three stages: carboxylation, reduction, and regeneration. Now, during carboxylation, what happens? A molecule of ribulose 1,5 bisphosphate. So, the short form of this is RUBP. So, during carboxylation, a molecule of ribulose 1,5 bisphosphate combines with the molecule of carbon dioxide to form a molecule of 3-phosphoglycerate. 3-phosphoglycerate. So, ribulose bisphosphate combines with carbon dioxide to form 3-phosphoglycerate, that is 3-PGA. Okay. Now, this reaction, okay, is catalyzed by an enzyme called Rubisco. Rubisco is the most abundant biomolecule in the entire biosphere. Okay. In the next step, that is reduction, this 3-PGA is converted to a triose phosphate. Okay, in reduction, 3-phosphoglycerate is converted to a triose phosphate. Now, this reaction requires one molecule of ATP and one molecule of NADPH. Now, each triose phosphate is a 3-carbon compound and contributes one carbon. It contributes one carbon for the synthesis of glucose molecule. So, triose phosphate is a 3-carbon compound. One of the carbon from the triose phosphate is contributed towards the formation of glucose, and the remaining two molecules of carbon are used for, okay, regeneration of ribulose bisphosphate. Now, this regeneration step, it requires one molecule of ATP. Now, since glucose is a 6-carbon compound, it requires 6 Kelvin cycles to produce one molecule of glucose. Okay. Number of ATPs used in one cycle is 2. So, one ATP is used, okay, during the reduction process, and one ATP is used during the regeneration process, and one NADPH is used during reduction process. So, in a single cycle of Kelvin cycle, in a single Kelvin cycle, two molecules of ATP and one molecule of NADPH are utilized. Hence, it requires 12 ATPs, 6 NADPH, and 6 carbon dioxide molecules for the synthesis of one molecule of glucose. This cycle is also called as C3 cycle because the first stable compound, here you can see the first stable compound in this cycle is 3-phosphoglycerate. 3-phosphoglycerate, which is a 3-carbon compound. That is why this cycle, Kelvin cycle, is also called as C3 cycle. So, this is the balance sheet of the Kelvin cycle. So, total input, okay, so this is input, this is output. So, in this reaction, what happens? Okay, in case of Kelvin cycle, 6 carbon dioxide molecules are utilized for the formation of one glucose molecule. So, each carbon dioxide, it contributes one atom of carbon for the synthesis of glucose, and glucose is made up of 6 carbon atoms. So, 6 carbon dioxide molecules are utilized, and also 18 ATPs are utilized for synthesis of one glucose molecule. In each cycle, 3 ATPs are utilized. So since there are 6 cycles, 18 ATPs are utilized, and the end products are 18 ADP. Similarly, 12 NADPH are used. 12 NADPH are used. Actually, this step requires 2 NADPH and two molecules of ATP and one molecule of NADPH. Okay. Now, I think this we are not explained properly. Okay. Remember that for the reduction purpose, in each cycle, two ATPs and two NADPH are utilized. Means in a single cycle, in a single Kelvin cycle, 2 plus 1, 3 ATPs are used, and 3 ATPs are used, and 2 NADPH are used in one cycle. So 6 cycles means 12 NADPH molecules. So for synthesis of one glucose molecule, it requires 6 carbon dioxide molecules, 18 ATP molecules, and 12 NADPH molecules. And the outcome is 1 glucose. These are the products: 18 ADP molecules and 12 NADP molecules. Now, let's discuss about C4 pathway, C4 cycle, or which is also called as Hatch and Slack pathway. This is observed as an alternative to C3 cycle, C3 pathway in some plants called C4 plants. The most common examples for C4 plants are maize, sugarcane, sorghum. Sorghum is nothing but jawar, and also millets. Now these are all C4 plants. Now C4 plants show a very special anatomy of leaves called Cran's anatomy. Here, the bundle sheath cells are in close association with the mesophyll cells. Now, this is the bundle sheath cell, okay, that is found around the vascular bundles, and this is the mesophyll cells. Now, they are found in close association. So, this is the mesophyll cell, and this is the bundle sheath cell. Okay, so bundle sheath cells are in close association with mesophyll cells. Now, both bundle sheath cells and the mesophyll cells are involved in the synthesis of carbohydrates. Now, this is the representation of C4 pathway. This is the mesophyll cell, this is the bundle sheath cell. Now, C4 cycle is completed in 4 basic steps. The first step is fixation, then decarboxylation, and fixation of carbon dioxide by Kelvin cycle, and regeneration. Now, atmospheric carbon dioxide, it gets dissolved, okay, in the cytoplasm of the mesophyll cells as bicarbonate ions, HCO3. Okay. Now this HCO3 combines with one molecule of phosphoenol pyruvate. Okay, in short it is called PEP. Okay, HCO3 combines from PEP, that is phosphoenol pyruvate, okay, which is a 3 carbon compound, okay, to form a 4 carbon acid, okay, such as oxaloacetic acid. Now, since the first stable compound in C4 pathway is a 4 carbon acid, it's a 4 carbon acid, that is why it is called C4 pathway. In C3 pathway, the first stable compound is a 3 carbon compound, that is 3 phosphoglycerate, but in C4 pathway, the first stable compound is a C4 acid such as oxaloacetic acid. That is why it is called as C4 cycle. Now this C4 acid is then transported into the bundle sheath cells. Okay, remember Kelvin cycle in case of C4 plants occurs in bundle sheath cells, not in mesophyll cells. In C3 plants, Calvin cycle occurs in mesophyll cells, but here Calvin cycle occurs in bundle sheath cells. Okay. Now this C4 acid, that is oxaloacetic acid OAA, this C4 acid this C4 acid is transported into the bundle sheath cells. It is transported into the bundle sheath cells. Okay. Now this C4 acid undergoes decarboxylation to release carbon dioxide and a 3-carbon acid. Okay. Decarboxylation process. In decarboxylation process, what happens? C4 acid is converted into C3 acid and carbon dioxide is released. Now carbon dioxide thus released enters into the Calvin cycle for the synthesis of carbohydrates. Okay. This C3 acid is then transported back to the mesophyll cells and it is used, this C3 acid is used in the synthesis of PEP, that is phosphoenol pyruvate, which is ready to be combined with HCO3 to form C4 acid again in the next cycle. Now, one more interesting event occurring in C3 plants, which is absent or very rare in C4 plants, is nothing but photorespiration. Okay. Now, to understand photorespiration, we have to understand the nature of the enzyme Rubisco. So, you know very well that Rubisco is very important. Okay. So, this Rubisco means ribulose bisphosphate carboxylase oxygenase. Carboxylase oxygenase. So this is nothing but Rubisco. Now this enzyme, what normally it does, it combines with carbon dioxide and ribulose bisphosphate to form 3-PGA, that is 3-bisphosphoglycerate. Okay. Now this 3-bisphosphoglycerate, it contributes carbon for the synthesis of glucose in C3 pathway or Calvin cycle. So this is the normal reaction. Now the enzyme Rubisco has dual affinity. It has affinity towards carbon dioxide. It also has the affinity towards oxygen also. Now during photorespiration, Rubisco combines with oxygen and RUBP to form phosphoglycolate. Now this reaction is nothing but photorespiration. This reaction does not produce any carbohydrate, but also makes use of ATPs. So it is a wasteful process. Photorespiration is absent in case of C4 plants. Now this is possible because C4 plants have a mechanism to increase carbon dioxide concentration. This increase in carbon dioxide concentration makes Rubisco to develop more affinity towards carbon dioxide to avoid its binding with the oxygen.